Okay, so hi everyone. So now we are going to do our last talk of this series entitled Understanding Bat and How to Phenotype Mouse Models for It. So this is a little bit different to our previous talks because it's kind of quite a distinct set of phenotyping protocols you need to go through if you want to look at thermogenesis and study the processes of how the body makes heat as opposed to processes sort of regulating things like energy balance and carbohydrate and lipid metabolism. And for that reason, I'm going to start at the end with our conclusion slide. And so what we can do is we can think about these boxes that we have here split into different sections as showing components of things that make up metabolic rate. So the components contributing to overall metabolic rate. And at 24 degrees C, we will have a metabolic rate that's yay big. And this will be increased by going to 5 degrees C. But I've shown some different states to indicate that the components that go to making up the metabolic rate can be quite different. So we'll always have a component of basal metabolic rate. So this is just the energy expended by the tick over of your organs, because you're going to require energy for that. But then you're also going to have components such as shivering or muscle non-shivering thermogenesis is another option. And then you have a noradrenaline dependent part if you are working at subthermoneutral temperatures, and this is 24 degrees C, of which the majority will be BAT mediated and some of it will be non-BAT mediated. If you move from the fasted to the fed state, you may start to see other things like the thermic effect of feeding coming in. And then if we move to the cold, you will get a big increase in metabolic rate, but that will not be involving the same components at the same time. So acute cold exposure is heavily driven by shivering and potentially some muscle non-shivering thermogenesis. It's still a bit controversial if this really exists, as well as some BAT and your BMR. Chronic cold adaptation will lead to a switching off of shivering and a much larger expansion of the BAT and noradrenaline dependent component. And then genetic modifications such as deleting UCP1, which we'll get onto in a moment, which is necessary for BAT to function, that's going to lead to a huge reliance on shivering and a loss of this BAT-dependent process. Now, what determines the difference in metabolic rate between 24 and, say, 5 degrees C? Well, this differential is really going to be determined and controlled by your brain responding to the environmental cues that it has, both external in the form of temperature, but also internal cues such as core body temperature, hormones, and various other factors. And we're going to talk through how this impacts on BAT and our understanding of brown adipose tissue. And as I mentioned already, we can have the same high metabolic rate, but it can involve very different components of what is causing the energy to be expended within the body. So how to phenotype for BAT? We're going to talk about the relevance of free living calorimetry, and the short story is free living calorimetry is not a good readout for thermogenesis and certainly not for BAT activity. So that's going to take up probably about the first half of our talk is about how not to phenotype for BAT. But I think it's important to understand this because it gets misconstrued a lot in the literature. Just because your mouse is showing hypermetabolism, something we covered in the first lecture, it doesn't mean it's caused by BAT. Then we're going to talk about thermogenic capacity and how we measure that. It's also using calorimetry, but we have some specialized conditions that enable us to measure BAT. UCP1 levels in terms of mRNA and protein and what they mean, BAT weight and its lipid content and what that might mean, a tiny bit on other molecular markers focusing on mitochondria because I think it's got some interesting points that we can make from that. And then finally, we're going to look at measuring really whether the brain is signaling to BAT for BAT to be turned on as part of that component of the metabolic rate. Okay, so what is BAT? So most people will know, but brown adipose tissue is a specialized organ that turns energy into heat. And it's necessary in small animals and infant humans to have lots of BAT in order to keep warm. So this organ's sole purpose is to make heat. Histologically, it looks like this. BAT differs from WAT in that WAT is made up of large unilocular, as in they have one lipid droplet, adipocytes, whereas BAT has many smaller adipocytes with lots of little lipid droplets. Bat is found in mice, but also we now recognize that it's in adult humans. In mice, the largest single depot is located between the shoulder blades, the interscapular region. In the human, the largest depots of bat are located in the subclavicular region and about here. Now, what is different between bat and wat is obviously we have these giant lipid droplets, but in bat, we have these little lipid droplets and a lot of this red staining from the H&E stain. And that red staining is staining predominantly mitochondria. The cytoplasm of bat is chock full of mitochondria, and mitochondria are essential for bat's function because to produce the heat, it needs to do oxidative phosphorylation. And the way bat turns energy into heat is it performs oxidative phosphorylation, and it uses the electron transport chain in the way any mitochondria does to pump protons into the membrane space. But it doesn't use ATP synthase then to generate ATP. Instead, the protons pumped by the ETC just dissipate down the inner mitochondrial membrane, and the byproduct is heat. And it's very analogous to if you short circuit a battery with a piece of wire from the positive to the negative terminal, that wire will get hot. And the same process is happening here. And so here's an animation I once made showing ATP synthase and the inner mitochondrial space, the matrix here, and protons going through UCP1. And UCP1 is the uncoupling protein. So this is what allows the flow of protons through. And what is different about UCP1 to other uncoupling proteins that have been suggested to maybe be able to allow protons to flow through, and also a concept of basal metabolic leak, is it's highly regulated, its activity. It's switched off by the binding of purine nucleotides, and it is switched on by the binding of fatty acids. And this means you can very acutely turn on or off heat generation. And that's super important given our earlier talks about the low heat capacity of a mouse. You don't want to go from a cold environment with lots of heat production to a warm environment, and then the mouse cooks itself to death because it can't turn off its heat production. So how is this activity of bat controlled? What is the principal signal regulating whether brown fat is active or inactive? The answer is the brain. And there's a complex neurocircuitry. This is from Sean Morrison. I should have put his paper heading up here, so apologies to that. I think I've done it with most of the papers. And there are really two major sensory inputs, two neuronal inputs, that are going to set the tone ultimately down here for the signal going to IBAT. They are temperature sensing neurons in the skin, which have their nerve bodies in the dorsal root ganglia. They go to the dorsal horn through some areas of brainstem and into the hypothalamus, into the medium preoptic area. In the lateral hypothalamus and the medium preoptic area, there are also some warm sensitive neurons that are actually sensing the temperature of your brain. So you are able to integrate in the preoptic area the signals from the environmental temperature outside as well as your core body temperature. And we're going to see these are really important. This then leads to an outflow via another series of brain regions all the way down here to where ultimately sympathetic neurons signal to brown adipose tissue, and the main neurotransmitter that is signaling to brown adipose tissue is norepinephrine from sympathetic neurons. Now, bat is a highly plastic organ in terms of its mass, its lipid content, and its thermogenic capacity. I'm going to revisit these images later, but I just want to show here, if you have a mouse at 28 degrees C, this is showing all the adipose tissue depots in the mouse. Now, this is, I'm almost certain from Saverio Cinti's laboratory. I couldn't find the original paper, but I did find the image in Google image search. And what it shows here is this large bat depot, which is found between the shoulder blades, and these other large depots which are found located under the shoulder blades. But most of the other fat is predominantly wat, and this is at 28 degrees C. At 6 degrees C, almost all of these fat depots have to some extent gone white, with the least whitening going on in the perigonadal depots and also in the omental white fat. The lipid content of bat, those lipid droplets we saw in our H&E image of brown adipose tissue, is also incredibly plastic. It takes only 12 hours of moving an animal from room temperature to 4 degrees C to literally remove all the lipid. They will become completely delipidated. And then over the next 24, 48 hours and then 7 days, they will slowly restock their lipid as DNL processes catch up and lipid intake catches up to match lipid oxidation. So this browning is caused predominantly, we think, by the recruitment of new brown adipocytes, and also within the existing brown adipocytes themselves, there is also an induction in the amount of this uncoupling protein, UCP1, this critical proton, regulated proton leak molecule necessary for the heat production. And the total amount of UCP1 in the brown adipose tissue of the organism matches very well with thermogenesis. So on the x-axis, we have temperature here. And as environmental temperature drops, the demand for heat production increases, and you will see a linear relationship between oxygen consumption or energy expenditure with temperature, and you will see a linear relationship between the total amount of UCP1 in the animal and the temperature. And this is on protein. And another point to note here is this here above about 28 degrees C up until about 32 to 34 degrees C, something like that, is the thermoneutral zone. And at this point, the animal is not producing any heat or expending any energy for the purpose of regulating its body temperature. It is when you drop below this thermoneutral zone, you see a very rapid increase in oxygen consumption, and that is bat dependent. So you can see by the time we get to 5 degrees C, well over 60% of the energy in the mouse is being expended for the purpose of thermogenesis, and the vast majority of that is in bat through this UCP1 protein for essentially allowing direct heat production through the uncoupling of the electron transport chain. There is obviously some additional energy for things like powering the heart, because you're going to need to pump an awful lot more oxygen and a lot more food in the blood around the body, but that's also pretty much for the purpose of generating heat, so that kind of adds in with that. Now, the molecular mechanisms regulating BAT, we could have a whole conference about the transcriptional systems that go on to regulate BAT. But for the purpose of this talk, we're only going to talk about one thing, and that is norepinephrine. And I would argue that that is the single most important signal. So this is a figure I have cribbed from Brown Adipose Tissue Function and Physiological Significance. This is a review from, I think, 2004, written by Barbara Cannon and Jan Nedergaard. It's been cited 4,500 times and is also known by many in the BAT field as the BAT Bible. It's a fantastic resource if you want to know about brown adipose tissue. Even though now it's pushing 16 years old, it's still superb. Anyhow, norepinephrine, in terms of BAT, does two things. It promotes both the differentiation and recruitment of new brown adipocytes. So when we saw that 28 to 6 transition, the conversion of that white fat to brown is at least in large part dependent on the presence of functional norepinephrine signaling, and that's acting through beta-1 and beta-3 receptors. The beta-3 receptors are thought to be more responsible for the activation, telling UCP1 to turn on. But both processes are implicated. Norepinephrine binds beta-3, it's GS-coupled receptor, it activates adenylate cyclase and raises cyclic AMP, which in turn activates PKA. That will activate lipolysis and drive thermogenesis, but it will also activate CREB, and CREB will drive the thermogenic program, for example, increasing the expression of UCP1. It takes time, though, for BAT to become fully recruited. So if you take an animal from 28 degrees and 6 degrees, this conversion from lots of white fat to predominantly brown and beige fat does not happen instantaneously, and it happens over a course of, we think, about 4 weeks. And so what you will see in this panel, which is from this review, UCP1 mRNA does not produce heat, again from Jan Nedergaard and Barbara Cannon, is the first thing that will happen immediately is that total metabolism will go through the roof. Because if the mice do not expend energy, they will become hypothermic and die. However, their BAT is not fully recruited. And if their BAT is not fully recruited, they do not have enough BAT capacity to make all that heat. So they have to do it another way. And the way they do it is with shivering. So just like we would on a very cold day start shivering, the mice will start shivering. But this degree of shivering will decline over time as it ceases to be needed, because the non-shivering thermogenesis capacity, this recruitment of BAT, increases over the 28-day period, concomitantly with the total UCP1 amount, until you have enough thermogenic capacity that you no longer need shivering in order to maintain this high metabolic rate. So we can think of this whole system as kind of analogous to a car engine and accelerator. So the brain is like the driver integrating all the signals, so what they're seeing happening on the road, to decide how fast they want the car to go. And then they're going to press the accelerator pedal, which is this connection here, which is the S&S, and that's going to go to the engine, and it's going to make the engine either go faster if you press the accelerator harder, or if you take your foot off the accelerator, the car's going to slow down. So the conclusions of this little introduction are that BAT is a highly plastic organ capable of expanding and contracting in size in response to environmental cues. It generates a huge amount of heat in mitochondria for the purpose of thermal homeostasis. It becomes more active at lower environmental temperatures and can account for over 60% of all the energy expenditure in a mouse. The main organ controlling BAT activation is the brain, and it does so via the sympathetic nervous system. And the key neurotransmitter regulating BAT is noradrenaline. I think that these are fair statements. There is obviously nuance to all of this. So what are the physiological cues that regulate this differential between 5 degrees and increasing it or decreasing it down to 24, increasing it up to 5? What are our signals that are going to mean we have high metabolic rate and high BAT activation? So I've already alluded to them, but there's basically 2 major factors. And the first is this: it is the gradient in temperature between the mouse's core and the environment. And the colder the environment is, then the greater this gradient, okay? And we're also going to see if you alter the mouse's core body temperature, that can also affect this gradient. The second is insulation. So if a mouse is well insulated, it will dissipate less heat to the environment and need less heat to maintain its core body temperature. If it is poorly insulated, it will dissipate more heat to the environment and it will need more energy in order to maintain its core body temperature. So we're going to go through the factors. We're going to go through environmental temperature, core temperature, and insulation to show how they impact on energy expenditure and also, at the end, brown fat. So environmental temperature. We've already seen this, and this is something from my own work. It's just looking at the relationship between environmental temperature at which you're housing your mice and increment in energy expenditure of the mouse, and in this case, it's expressed in watts. So that's pretty straightforward, but this one I think is slightly more interesting, which is the core body temperature of the mouse. Because if the core body temperature reduces very much like this, 1 degree here is worth about 6% energy expenditure, which is quite big. So, and you can alter your core body temperature by between 0.5 and 1 degree, so it can have an important effect. So the first thing is we can just look at a natural circumstance of mice which have a higher core body temperature, and this is just simply looking at the difference between males and females. In part because of the estrous cycle, females tend to defend a core body temperature which is on average between 0.5 and 1 degree higher than male mice. So we took a load of data from calorimetry, this is from the Wellcome Trust Sanger Institute, for male and female mice for the high-fat diet and also chow males subsequently. This is what they have. They have not put chow females through. And these are all wild-type mice. But what you can see is the metabolic rate corrected for ANCOVA is similar between males on a chow and a high-fat diet, at least after they've been on a high-fat diet for 12 weeks. But for the females, it was significant, slightly and significantly higher. And I went looking for this in the literature this morning, and I found a few papers on it, but this one seemed the cleanest. In blue, we have females. This line here is core body temperature. And in red, we have males, and this line here is core body temperature. And you can see on the y-axis, the difference is a little under 1 degree in their core body temperature, probably about 0.5 degree on average. And so that's pretty much enough to explain that increased metabolic rate, because even though the mice are being phenotyped in a room that's 21 degrees, whether they're male or female, the gradient between their core temperature and the environmental temperature is about half a degree larger for the females, so they need a little bit more energy to maintain that higher temperature. It's also possible to alter core temperature genetically. So this was a really nice study from the lab of Thomas Bartfai, and what they did was they targeted those neurons I mentioned in the lateral hypothalamus and the preoptic area that sense temperature. And the way they targeted them was they decided to make those regions of the brain hotter by virally overexpressing UCP2. Now, UCP2 is an uncoupling protein, but if you express enough of any inner mitochondrial membrane protein, you will render the inner mitochondrial membrane slightly leaky. And if you render the inner mitochondrial membrane slightly leaky, you will get protein leakage and you will produce heat. So whether it was truly uncoupling or simply punching holes in your mitochondrial membranes, the net result was they increased heat production very specifically in these two nuclei within the hypothalamus, and it was sufficient to increase the temperature of those nuclei in the hypothalamus by about 1 degree. So changes in temperature tend to not be very large, because if they'd increased it by 3 degrees, they'd have cooked those regions and killed all the cells in that area of the brain. So you get about a 1 degree increment in temperature. And the result was the mouse's brain is tricked into thinking that the mouse is too hot. So what does it do? Well, it reduces core body temperature. So the red lines are the transgenic animals. And also, as a marker of energy expenditure, they fasted the mice and asked how much weight did they lose. And the mice defending a lower core body temperature, and again, we're looking about half a degree, lost less weight in a paradigm where they had no food. So the differential is strongly driven by energy expenditure. They didn't have calorimetry in this experiment. So we will conclude, if the temperature gradient between the mouse's core temperature and the environment increases, energy expenditure will go up. The gradient can increase either because the environmental temperature changes or because the core temperature changes. So the next thing we're going to look at is insulation. So this was a nice paper by the ex-head of the DMC, Amy Warner. And what they did was they looked at a thyroid receptor alpha 1 mutant. And this mutant was interested because it had a low core temperature, but that low core temperature was paradoxically, apparently, associated with an increased energy expenditure. So this is the temperature, the black are the mutants. This is the energy expenditure, the black are the mutants, and really quite a large one. So this seems strange because you would expect high energy expenditure to increase core body temperature, or a low core body temperature to require a lower energy expenditure. But it turned out what was happening was the thyroid receptor mutant was affecting the vasculature, and it was causing vasodilation in the tail. And the tail is a major thermal regulatory organ in mice. And as a result of greater vasodilation, the mice were losing more heat to their environment. They had more bat activation as a result of their requirement to generate more heat. So the brain was telling their bat, we're cold, turn on. And despite having a big increase in energy expenditure, they still were defending a slightly lower core temperature. So this shows that insulation can actually have a big impact on energy expenditure, and that the body temperatures may not be such a great readout for understanding thermogenesis, because you have low body temperature and high thermogenesis. Another thing that can happen is less physiological regulation of your temperature. So vasodilation is a physiological mechanism for regulating your core body temperature. And that is, you can have skin barrier dysfunction. So SCD1, steroid-CoA desaturase, is essential for producing lipids that are found on the surface of the skin of mice. And when you lose it, you start to have hair falling out. These mice will eventually go pretty much completely bald. And they also have problems with their skin barrier in terms of the lipid layer that should coat all of our skin. Now, as a result of this, their skin becomes far less good at insulating the mice. And you get a truly spectacular increase in energy expenditure. It's a near 30% increase in energy expenditure. Now, the largest I think I've ever been involved in publishing is 8%. So this is a massive increase in metabolic rate. And it's being driven because the animals are losing so much heat to their environment, and they have to make more. Now, bear in mind, this is a deletion in skin. and yet we're seeing a huge increase in energy expenditure. Now, to prove that it really was heat dissipation, what they did was another group actually used total SCD1 knockout, which is the same phenomenon of hypermetabolism and messed up skin, and they slathered them with Vaseline. And by covering the mice in Vaseline, you can see the red is the knockout without Vaseline, the blue is the knockout with Vaseline, and by coating them in Vaseline and restoring some thermal insulation, they were able to partially normalize them back towards the wild type levels. So this is going back to the skin knockout of SCD1. So despite this being a gene exclusively knocked out in the skin of the animals, you saw a big upregulation of genes involved in thermogenesis in bat. So bat was, in this case, being the effector of the heat production. Increased UCP1, increased CPT and LCAD, which are responsible for beta oxidation. Increased PGC, which drives the whole thermogenic program, and increased lipolytic marker, HSL, increased beta-adrenergic receptor, which is the receptor that is the norepinephrine is signaling, and also this enzyme deiodinase 2, which converts inactive T4 to active T3 within the cell, and thyroid hormone is a very important regulator of brown fat. So decreased insulation increases heat loss to the environment, and this increases demands for heat production and therefore leads to bat activation and recruitment. So kind of based on these concepts, it doesn't seem that bat is the thing that regulates metabolic rate. It's the brain regulating the metabolic rate. So if we delete a gene in bat, would that cause hypermetabolism, hypometabolism? We would argue not. The factors that will determine your metabolic rate are your core temperature, your insulation, and the thermal gradient to the environment. So is this true? Well, the answer is categorically yes. You can actually eliminate UCP1 in mice and render your bat completely functionally null, and you do not alter the metabolic rate of your animals. So this is body temperature. These are UCP1 knockout and wild types that have been warm adapted or cold adapted, mild cold adapted 18 degrees. And if you transfer them from 30 degrees to 5 degrees C, you see absolutely nothing on the warm adapted, and you see virtually nothing on the mild cold adapted. And in fact, you can have UCP1 knockout mice live at 5 degrees C quite happily, and their energy expenditure is reported to be the same, or in some cases even maybe slightly higher than controls. What is different is their capacity for heat production. So this was a challenge where they tested the maximum energy or power that the mice could put out, so the maximum heat production. And in wild types, they could reach about 800 milliwatts in warm adapted animals. Cold adapted animals could reach about 1200 milliwatts. And UCP1 knockouts, first of all, could not survive at such low temperatures. So maximum heat production in a cold adapted mouse is at minus 18 degrees centigrade. Whereas in a UCP1 knockout, it's only at minus 10. Below that, they will die. But you can see here that this energy equation is passed into different sections. And you can see that the norepinephrine controlled section, which is heavily driving brown adipose tissue function, is way smaller in UCP1 knockouts than in wild types. And so so this gives us a clue as to how they're actually able to survive in the cold, because we showed this earlier from the UCP1 mRNA doesn't make heat, and this is showing what happens in a wild type mouse. If we consider a UCP1 knockout mouse, when we put it in the cold, well, what happens? Well, total metabolism has to go up or the mouse dies. However, over 30 days, shivering does not diminish, and this has been proven experimentally with EMG readings. Non-shivering thermogenic capacity really does not increase in the way it would in a wild-type mouse, and the total UCP1 amount obviously is completely flatlined. So you're still able to do the metabolism, but you're doing it using different components of that metabolic rate chart that we showed in the very first slide. So to look into this further, we sort of asked, well, maybe the UCP1 knockout was just a specialized case. And so we decided to see if we could find any genetic evidence that genes in bat aren't really responsible for the regulation of metabolic rate. So what we did was we went to the IMPC data, and this got published in Nature Communications. And interesting, one of the authors is the new head of the DMC, Chris Elliott. So again, a lot of props for the DMC in this. And what they did was they took thousands of lines that they'd phenotyped, and from those lines that they'd phenotyped, they had about 1,000 of them had gone through calorimetry. And they calculated metabolic rate correctly using a variation of ANCOVA. And then they plotted the normal distribution of metabolic rate in these animals. And they looked at the extremes. So they classed the top 5% of these lines as being hypermetabolic, the bottom 5% as being hypometabolic, and the lines that fell in the middle, 90% in the middle, as being eumetabolic. Okay? And so the argument we would make is if genes in bat, alterations in gene function in bat, is able to alter free-living metabolic rate, then what you would expect to see is that more of these hypo and hypermetabolic genes would be expressed in bat than the eumetabolic ones, i.e. they'd be overrepresented as bat genes. And we don't find that at all. We actually find that genes which, when you knock them out, render mice hypo or hypermetabolic are actually slightly, but significantly, less likely to be expressed in brown fat. This axis starts at 0.86 and goes to 0.905. So the difference is just 2%, but it does formally reach significance, I think, because these two have exactly the same ratios in this setting. And equally, we also found there wasn't any real regulation of these genes at all. So of the genes that were expressed in bat, they were no more likely to be regulated by, say, cold exposure if deleting them resulted in hypometabolism or hypermetabolism. So put simply, it seems that evidence from the UCP1 knockout mouse and also evidence from mouse genetics suggests that manipulating gene function in bat is not going to alter your metabolic rate. Doesn't mean it doesn't alter your bat function. The UCP1 knockout has no bat function, but it's not going to alter your metabolic rate as measured by calorimetry. Okay then, so this goes back to our idea that bat is the engine, our brain is what's controlling the accelerator pedal, and then the SNS is the cable connecting the accelerator to the bat. So the key point here is your metabolic rate is controlled at the level of your brain. It is not controlled at the level of your bat. So why do we care about this brown adipose tissue stuff then if it doesn't affect energy expenditure? Well, it can do loads of great things, particularly if we have a way to activate bat by bypassing the accelerator and just turn that engine on. You know, if you hyperactivate bat with cold exposure, you can basically cure dyslipidemia. This is an ApoA5 knockout mouse after just 24 hours of cold exposure. You can also cure NAFL. So this is an LDLR receptor knockout mouse. Again, 24 hours of cold exposure, you just strip all the lipids out of the liver. If you have a UCP1, a synthetic form of UCP1 called dinitrophenol, well, if you give this in a dose-response manner, from a dose of 0.1 gram per day to 0.8 grams per day across a huge range of patients, you will see a weight loss that ranges from 1 to 3 pounds in a week. So that's 1.5 kilos in a week, which is a spectacular amount of fat. It is dangerous because burning 800 grams of fat is about 7,200 kilocalories per week. A 30% increase in BMR is equivalent to 4,700 kilocalories per week. And the dangerous side effect of dinitrophenol is if you overdose on it, you will cook yourself to death because you raise your energy expenditure so high. Also, not having functional BAT can be very bad in the case of mice, and this is more relevant to us. So whilst those UCP1 knockout mice can survive in the cold, at least initially, they start dying. And actually, the most likely reason for them dying, though it was never worked out formally in this paper, but it's been looked at in some other research on slightly different models, is because they have to shiver continuously, their muscles start to break down, and they get massive amounts of creatine in their blood, and this screws up their kidney function. So you'll essentially start to go into a severe muscle damage state if you do not have functional BAT, because you're so reliant on shivering. And shivering is not a process that's designed to work for, well, in this case, 10, 20, or 30 weeks. Okay, so the conclusion of the first half of the talk is that free-living metabolic rate is not a great readout of BAT function, nor is it really regulated by changes in BAT function. And while intrinsic changes in BAT do not appear to impact on metabolic rate, activation of BAT has multiple metabolic outcomes which are beneficial. So if we can turn it on, it's a good thing. And loss of BAT can be compensated by other metabolic processes, but it's not sustainable in the long term. If you have no BAT, yeah, sure, in the short term for a few weeks, you can live at 5 degrees, but you're not going to be able to live at 5 degrees for very long. Okay, so now we're going to go back to our analogy of the car, and we're now going to go to looking at measuring BAT function and measuring how big our engine is the first one. So many of the tests we conduct to see how functional BAT is ask the question, what is the capacity of BAT for thermogenesis? And so the first one we're going to talk about is a calorimetric method called thermogenic capacity. Then we're going to just briefly revisit UCP1 levels at the mRNA and protein levels, because we've done that quite a lot. We're going to talk about BAT weight. You'll often see BAT being weighed and its weight reported in the literature. What does that mean? And then we're going to do a little bit about mitochondrial markers and a specialized case of that, because that's really a whole separate topic, investigating mitochondrial function. Okay, so how do we measure thermogenic capacity? So we have this specialized rig that is, as you can see, is homemade in our animal facility. And what it is, it's a box which enables us to maintain animals at 30 degrees, and it's hooked up to our calorimetry system. And so we have individual temperature, cage temperature monitoring, and these cages are nicely both at 30 degrees. We have a gas heat exchanger, which is a copper pipe, because the air coming in is room temperature. We want it at 30 degrees when it hits the boxes. And we have this temperature controlled chamber. Very sophisticated. It was built for virtually nothing. We had a piece of copper that was £2, some diaries for insulation, a metal trolley which was free from the facility, some old Columbus Oxymax cages we had kicking around, home thermometers, and then this commercial hot box we'd had for bleeding mice, so we repurposed it for doing this. And so the protocol we do, and it's important to note the difference between measurement temperature and acclimation temperature, is you take two groups of animals. So you might have 8 wild types and 8 knockouts in your first group, 8 wild types and 8 knockouts in your second group. We're only just going to look at comparison of wild types. And we acclimate one of the groups for 4 weeks to thermoneutrality, and we acclimate the other group for 4 weeks to cold, to 5 degrees C. So this is the adaptation period length that we talked about earlier. It takes about 4 weeks to fully adapt brown fat. So this is really going to be an assay to test how well adapted our brown fat is and how much capacity we've generated. At the end of the acclimation period, we anesthetize the mouse and place it in a temperature-controlled environment at 30 degrees C, because once we've anesthetized it, we have to maintain its core body temperature. And this is done regardless of its acclimation temperature. So we do the measurement at 30 degrees C, regardless of whether they were acclimated to 30 or acclimated to 5. We then measure the energy expenditure in this temperature-controlled environment to get the basal metabolic rate. Once we have the BMR, we're going to inject it with a maximal dose of noradrenaline, one that will fully activate all the adrenergic-dependent thermogenic processes in the mouse, and this is going to then be analogous to determining the top speed of the car. We are bypassing the accelerator and just pushing the engine as hard as it can go. And this is what the data looks like. So we are not relying on the core body temperature environment gradient or plus or minus insulation effects to regulate the energy expenditure. We're just bypassing it by injecting the neurotransmitter. Okay, so we get our basal energy expenditure, and that is slightly higher at 5 degrees C than it is at 30 degrees C, because as a result of the cold adaptation, we're going to have a bigger heart, we're going to have more vasculature, we're going to have more machinery to enable the organism to pump more blood and generate more heat. But BAT should be turned off. We then inject the norepinephrine, and the norepinephrine stimulation will cause an increase in energy expenditure. And it will do this at both 30 degrees and 5 degrees C. Because now at 30 degrees C, we should have largely shut down our brown fat and de-differentiated and lost UCP1, mostly. But there are still norepinephrine dependent processes like signaling, like the heart, for example, that will increase energy expenditure. At 5 degrees C, we've made all of our white fat brown, and we've got loads of UCP1. And so this differential between the norepinephrine stimulated energy expenditure from the 30-degree acclimated mice to the 5-degree acclimated mice is the BAT-dependent thermogenesis. To show you this in the UCP1 knockout, which obviously shouldn't be able to do this, so this is some data again from Cannon and Nedergaard. This shows mice adapted to 30 degrees. We do see a slight differential between a UCP1 knockout and a wild type following norepinephrine injection. But after mice have been adapted to 4 degrees C, this differential becomes massive. UCP1 knockouts do not greatly increase their energy expenditure over BMR, a little bit more than they did at 30 degrees, again related to this increased capacity in things like heart, but the wild types have now a huge differential. So this is entirely bat dependent. Okay, moving on to UCP1 and other molecular markers. So this is good because if we think about that thermogenic capacity experiment, we're, at least in the UK, because we have to use an anesthetic which is not good for recovery, so it doesn't interfere with SNS signaling, you need to dedicate 2 complete groups of animals to do the assessment. So it might be a good idea to find out if there are any markers in your mice that indicate they might have some alterations in bat function. And this is again coming from UCP1 mRNA does not produce heat. And what this paper shows particularly is that UCP1 mRNA levels are basically go through a biphasic regulation. So if you take your standard protocol of, say, I don't know, 500 or 250 nanograms of RNA, you do an RT on it, and you measure UCP1, UCP1 levels will actually peak at about 2 days. And then they will start to diminish back down as a proportion of total RNA signal. So the total RNA content of the brown adipose tissue will increase over this time, and the total UCP1 mRNA within the whole brown fat of the organism will go up by about 2 to 3 hours and then remain constant. But the relative UCP1 expression is going to be a bit higher after 4-degree acclimation, but not much. The real peak is at around about 9 hours to 1 day. Okay. Now, if we compare this to protein levels, what you find is something slightly different. First of all, protein levels drop as a proportion of all the protein in the brown fat initially, and then they climb back up to really not be dramatically different from 28-degree acclimated mice, as in arbitrary units per milligram of protein. However, over the same time course, the total protein content of the brown fat goes through the roof, and therefore the total UCP1 protein content goes through the roof. And total UCP1 amount and total thermogenesis have very similar profiles. Now, I've often had people ask me, but like, what exactly are you doing in order to get this value? Oh, this is just again showing the temporal kinetics. But so what exactly are we doing to get that value? Well, what you do is you run a Western blot, and this is from some mice I have with a very powerful disablement in brown fat. And you take the UCP1 band intensity as arbitrary units, or alternatively, you can get some recombinant UCP1 and use it as standard, so then you can calculate an actual amount of UCP1 if you want to be as precise as saying nanograms, for example. So you get the band intensity, and then you multiply it by the micrograms of protein you extracted per mg of tissue. So when you do your tissue extraction for your protein extraction, you need to weigh the amount of tissue you're extracting from, and then you need to know the volume of the buffer, and then you can calculate how many micrograms of protein per mg of tissue you had. So you multiply your arbitrary Western blot intensity by the micrograms of protein per mg of tissue, and then you need to know the weight of your brown adipose tissue fat pad that you extracted your protein from, and then you simply multiply by the weight of the fat pad in milligrams. And that is how you get your total UCP1 signal per fat pad. And you can see here's mRNA here. It's relatively similar. The big difference lies in these knockouts, which actually do show somewhat of an increase in response to high-fat feeding, whereas on an mRNA level, they look identical. The difference between the blue, which is the wild type, and the wild type fed a high-fat diet is actually slightly more dramatic on a protein level. It's about 4-fold as opposed to about 2-fold on the mRNA level. So this is a really good marker of the thermogenic capacity of drones. So conclusion would be direct assessments of thermogenic capacity or indirectly measuring UCP1 protein content in bat are excellent indicators of the capacity of bat for thermogenesis. And I would consider these to be the primary readouts of change in bat function. So this would be your first readout to determine, is there likely to be something going on in bat function? UCP1 protein content is a really good place to start. Bat weight is really not very useful. And we kind of alluded to this earlier, which was from this paper from Christophelet et al. And it was showing that just 12 hours of cold exposure was enough to remove all the lipid in brown fat. Now, in our hands, brown fat is usually about 40 to 50% lipid, and that means 12 hours of cold exposure is enough to roughly halve the weight of your brown fat. Another process that will cause delipidation of BAT is fasting. This is my own data, and you can see based on this was animals fasted overnight up to a total of 25 hours during this experiment. We were culling them over this time period. And what we saw was that every hour that these animals were fasted for led to a 23 milligram loss in BAT weight. And going back to this data I showed earlier with this UCP1 per fat pad, so this is a readout of thermogenesis. These were the tissue weights of these animals. And what you can see is between the wild types on chow and a high-fat diet, we see an increased BAT mass of about 50% relative to the wild type, which is associated with a 4-fold increase in thermogenic capacity. So in this case, we have increased BAT weight associates with increased thermogenic capacity. Conversely, between the knockout on a high-fat diet and the wild type on a high-fat diet, we also see a 50% increase in BAT weight relative to the wild type on a high-fat diet, coupled with a 3-fold reduction in UCP1 content and thermogenic capacity. And this is because this fat is largely becoming light white fat and it's expanding, but what's expanding is lots of unilocular white adipocytes. So this just shows that BAT weight can go up in states where BAT is activated, and it can also go up in states where BAT is disabled. So it's debatable how useful it is as a variable. Okay, brief aside about mitochondrial density and functionality. So there's a world of literature out there assessing mitochondrial function in BAT, and measurements can be made using things like Clark electrodes, seahorses, Orberuses. And I'm only going to mention one phenomenon because I think that we could have an entire technical seminar or 2 or 3 on the first 2 points. And it's the differential between nuclear and mitochondrial encoded transcripts or proteins for electron transport chain components. So this observation, I first noted this when we were working on this paper, and it was a paper regarding the enzyme Elevol-6. This is an enzyme that converts palmitate to stearate. But what we ultimately found was the mechanism was by regulating mitochondrial fusion, it was able to impact on brown fat function. And this was because stearate acts to regulate transferrin receptor 1, and downstream of TFR1 ultimately is ubiquitination of mitofusin 2. Anyhow, what we noted was that when we looked at the relative expression of different components, different complexes of the electron transport chain, so complex 1, 2, 3, 4, and 5, we found in the coal, so that's the gray, the dark and light gray bars, not the black and the white ones, all of the mitochondrial encoded transcripts, indicated by an MT here, were downregulated. So here we've got MTCO1, MTCO2, MTCO3, ATP6, ATP8, which is also mitochondrial encoded, except for complex 3. MTCYB did not change. Anyhow, but more seriously, whereas all of the nuclear ones were unchanged. So there was this reduction. So what's going on here? I mean, our mechanism is on fusion, not transcription. But it turns out if you pretty much do anything to mess up mitochondrial architecture, it can impact on transcription, because a lot of the processes that are necessary for mitochondrial transcription to work require the mitochondria to go through quality control processes to ensure the DNA is of good quality and for the mitochondrial transcriptional and translational machinery to work. And so if you mess up fission or fusion, or the mitochondrial DNA gets damaged, you will lower your mitochondrial encoded transcripts. And this exact same phenomenon was seen in humans by Rob Semple and Nuno Rocha's work on mitofusin 2 mutants. And what they noted when they did RNA-seq on the white fat from these patients with mitofusin 2 mutations, this white fat's located in scapula, it's a kind of a camel hump, was that all the mitochondrial genes involved in oxidative phosphorylation were massively downregulated, whereas all the nuclear oxidative phosphorylation ones were all upregulated. And I mean, like, this coloring system is fold chained from -15 to +15. These were massive changes. So it's as though the nuclear system is trying to compensate for its loss of functional mitochondria by upregulating these oxidative phosphorylation genes. It doesn't work because the mitochondria aren't functional. So measuring mitochondrial gene expression and/or protein can provide you a nice quick readout about the following: how much mitochondrial electron transport chain capacity you have, which could add on nicely to the UCP1 data, but it can indicate issues with mitochondrial function, particularly if you've got a divergent regulation of mitochondrial encoded genes versus nuclear encoded genes. But it doesn't actually tell you if that's because of alterations in the ratio of mito to nuclear genes being caused by transcription, caused by DNA copy number, DNA damage, or fission or fusion events regulating these things. Okay, so we're nearly done. We're just going to focus a little bit on this area, the brain and SNS outflow to BAT. And so what we want to do in this case is, if we do an acute intervention which leads to an alteration in metabolic rate, how do we determine whether BAT may be implicated in that? Okay. And so we're going to do one about measuring body temperature and what information we need to interpret any changes in body temperature and assign those changes in BAT or any other thermogenic organ, but also measuring SNS tone, which will tell us if that signal, our connection between our accelerator pedal and our engine, has been activated. And therefore, it's very likely if we have increased SNS tone to BAT, we have increased BAT activity. Okay, so as I mentioned, body temperature is not set by BAT, but changes in body temperature may reflect BAT activity. But if we want to know whether BAT is important for our change in body temperature, we actually need to measure two things. We can't just measure body temperature itself, we also have to measure BAT temperature. And the critical point about needing both these temperatures is as follows. If body temperature changes before BAT temperature does, or concomitantly with BAT, then a primary change in BAT activity to drive that body temperature is unlikely. It is likely that BAT has got warmer or colder because the core body temperature has got warmer or colder. Conversely, if the BAT temperature changes first and then the core body temperature follows, it indicates that BAT is likely the primary effector. And just to demonstrate this is some really nice work from Clémence Blouet that we were involved in a bit. What it's showing here is BAT temperatures, which are the diamonds, and core body temperatures, which are the squares. You can see BAT temperature is initially slightly warmer than core body temperature, which makes sense because it's a heat-producing organ. But given that the environment at room temperature, BAT's producing about 40% of all the heat in the body, it's only 1 degree higher on account of the spectacular vasculature of the BAT. So BAT can't get super hot because it would cook itself and die, but it does run about a degree hotter than core body temperature in these measurements. Anyhow, Clémence was studying a bunch of neurons which, when they got activated, turned off, we believe turned off BAT, but they certainly reduced core body temperature and they reduced metabolic rate. So one of the reviewers asked for this, and what the experiment is you have a thread which is able to either activate or turn off the neurons. I can't remember which one round it is. And when you add a drug called clozapine N oxide, it activates this receptor and either hyperactivates or silences the neurons. Anyhow, following the injection with clozapine N oxide, what happens is you see a dramatic reduction in both core temperature and BAT temperature. Whereas saline, you continue to see this nice, fairly reasonable maintenance of the temperatures. And you'll also note you maintain this nice 1 degree difference between the BAT and the core temperature. However, if we zoom in on this region here, what you can see is that the diamond, the BAT temperature, drops first. And it drops rapidly to come to the core temperature, and then the core temperature follows on down after 15 minutes. So this is showing the temporal kinetics demonstrate BAT temperature is changing first, strongly indicating that a reduction in BAT activity is the thing that is driving the reduction in core body temperature. You could, of course, do it a lot more directly. You could attach electrodes to the sympathetic nerves that subtend BAT, and it's absolutely beautiful if you can do this. So it can be done in unconscious animals, or it can be done actually now with telemetry, but I'm not aware of anyone who's got telemetry up and running for bat. Telemetry is certainly used for recordings of things like kidneys and adrenal glands. But what you do in this experiment, this is again from our lab's work, this is Andy Whittle's BMP8B paper, is they demonstrated that there was a difference between the wild type and the knockout's response to cooling, and that as you cooled the animal and dropped its core body temperature from 37.5 to 32.5 degrees, and this is in anesthetized animals, the electrode recordings showed differential SNS activity in the knockouts versus the wild types. So this is really nice evidence that you are actually having an alteration in sympathetic outflow as a result, in this case, of your gene modification. And this is just showing some of the recordings. You get these raw SNA spikes of firing, and then you can integrate them to look at your SNS tone, so how much firing you've got. And things like cold exposure will accelerate these. So that's great, but you need about £30,000 worth of equipment, and you need to be able to do microsurgery, and you also need to potentially have the whole surgical rig set up and only be able to do it in unconscious animals for some organs. What you could do instead is a much simpler experiment, which is to measure norepinephrine turnover by inhibiting tyrosine hydroxylase. So as neurons fire, they release noradrenaline at the synapse. And although some of this noradrenaline is reuptaken into the synapse, not all of it is. And the lost noradrenaline is replenished by the conversion of tyrosine to noradrenaline through this pathway, from tyrosine via L-DOPA, dopamine, to noradrenaline, and in some cases, ultimately to adrenaline. The first step in this synthesis pathway involves the conversion of tyrosine to L-DOPA by tyrosine hydroxylase. And if you inhibit tyrosine hydroxylase using alpha-methyl-P-tyrosine, then the noradrenaline cannot be replenished, and the noradrenaline levels in tissues diminish proportionate to how much their nerves are firing. Now, in contrast to our highly expensive surgical rig, this is the protocol. You take a mouse, you inject the mouse with alpha-methyl-P-tyrosine, you wait or do your intervention, we're going to see one in a second, you then kill the mice and harvest the tissues, and you measure the norepinephrine in the tissue, and you might normalize it per mg of protein if you wanted. And this is just to show some data from the effects of having mice at 18 or 30 degrees. And what you can see is the norepinephrine turnover here over 6 hours by plotting the log norepinephrine concentration on the y-axis against time. And you can see at 18 degrees, we get a steeper slope, as in we're losing more norepinephrine, and that slope is indicative of higher firing. In this case, we're looking at minus 0.28 versus minus 0.08. So we're looking at 3 times more SNS outflow to bat in this case, at 18 degrees than 30 degrees. And each of these points, I think it's about 3 or 4 animals, but this was a pilot study to see if we could get to work. Okay, so the conclusions from this section are the kinetics of body temperature change, as well as more direct measures of sympathetic tone to brown fat, can provide information about if BAT is involved in changes in energy expenditure within your mouse. And just going back to our very first slide, this differential here is metabolic rate. That is controlled by the brain and is determined by the environmental gradient, how warm and hot it is between the core body temperature and the environment, as well as the degree of insulation. So this level is determined by those factors. However, you can have very different amounts of BAT activity, shivering activity under different conditions. And these different events can have profoundly different outcomes for your mouse. For example, at 5 degrees C, after 30 weeks, almost all of these mice would be dead, whereas these ones would be very healthy and very happy. So this is the end. The overall conclusions for the seminar series are: we have covered energy balance and metabolic rate, food intake, RER, stable isotopes in mice, carbohydrate and lipid tolerance tests, and BAT phenotyping. And it's been a whistle-stop tour. There is much, much more to learn in the literature. I think the only thing I would really say to you at this point is I would encourage you to time travel. Much of the best stuff on animal physiology was published before 1990. And 1990 happens to be the invention of genetically altered mice. And so if you're really interested in physiology and phenotyping, I would strongly advise you go back, look at the literature, look at the hand-drawn figures, and then you're going to be able to learn a load of wonderful stuff about physiology.